The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
Let's delve into what we came to do today, glycolysis. So it's a metabolic pathway that actually breaks down or degrades glucose into pyruvate. So what I want to mention in this case is, in glycolysis you have a 6-carbon sugar usually. So we're going to be talking about glucose the entire time. That's a simple sugar that we're going to focus on. Now glucose is a 6-carbon sugar. So in the molecule, in the process of glycolysis, that 6-carbon glucose molecule is actually broken down into 2 molecules of a 3-carbon pyruvate. So you have a 6-carbon compound. A 6-carbon glucose is broken down into 2 3-carbon compounds, which is the pyruvate. Now glycolysis occurs almost exclusively in the cytosolic interface, and it results in the net gain of 2 ATP molecules. Now that is what I referred to earlier on as substrate level phosphorylation. It happens anaerobically without the input of oxygen as a consequence of the breakdown of that glucose molecule. Now, let's talk about something really interesting here. The fact that glycolysis actually happens without the input of oxygen tells us something really interesting. And I'm talking about evolution here, okay? I want you to stay with me. Because of the simple fact that glycolysis does not require oxygen, that would lead us to infer, to believe that it's probably a process that evolved early, very early in the history of life. And when I mean early, I mean before the formation or before the emergence of photosynthetic organisms that actually produced or released oxygen into the atmosphere. Remember that oxygen wasn't always present, right? So the fact that a process like glycolysis evolved without the use or without the input of oxygen, when several other processes, more specifically the next process, is dependent on the presence of oxygen, leads us to believe that it's a process that actually developed really early on in the history of life to produce energy without the input of oxygen, because oxygen wasn't there, right? Think about that for a moment. It's really, really interesting to try and contextualize some of these things from an evolutionary perspective at times. Now, back to what we're talking about. So for each molecule of glucose that goes through glycolysis, a bunch of things are produced. I've already told you that we produce 2 pyruvates. Okay, we also produce 2 ATP molecules, like I just said right here. A net gain of 2 ATP molecules, substrate level phosphorylation. And we also produce 2 NADH, that powerful reducing agent right there. So those are the main products of this process. It wouldn't hurt you to commit those to memory. It's really important to know what the net gain or the net products produced in this process are. A lot of questions are centered around that key concept, or around knowing that key piece of information. Now, glycolysis in and of itself actually involves a sequence of about 10 separate reactions. And we can divide these reactions into 2 distinct phases. So the first phase would be the investment of energy, or the phases or the stages of the reaction that require ATP, or that require the input of ATP. And in this process, we actually use 2 molecules of ATP per molecule of glucose. Now in the second phase, we can call it the energy return or payoff phase, is when we actually gain an additional 4 ATP molecules in the process. So that's why we're talking about a net gain of 2 ATP. The process of glycolysis will use 2, but give you back 4. So you have a net gain of 2 ATP molecules. Now the entire process is strictly controlled by enzymes. It's under enzymatic control, as you're going to see in the subsequent slides. So it's very easy to manipulate that process by controlling the activity of each and every one of those enzymes. So what we have at the bottom of the screen right there is basically glycolysis in a nutshell, right? This is glycolysis down here. So we have our glucose, and the input of the entire process is going to utilize 2-NAD nicotinamide iodine dinucleotide, that's in its oxidized form, 2-ADP, and 2 inorganic phosphate groups right there, which are going to be converted into 2-ATP. So this inorganic phosphate, these 2 molecules of inorganic phosphate are going to be coupled to the formation of 2 ATP molecules, and similarly that NAD will be reduced to form 2 molecules of NADH, the dehydrogenase form of the molecule. Water is also going to be produced as a byproduct. So what we have to the right of the screen right here, those are the key products. Those are the products of glycolysis as a process. So with a net gain of 2 ATP, we have 2 NADH, and we also have 2 pyruvate from each molecule of glucose. So specifics, let's delve into it. So I already told you it's a 10-step process. Step 1 through to step 10. Let's go over each and every one of them in a very detailed way. So